Well, hello everybody, and welcome to this Research in Action session on Scivision. Today we will have a set of presentations. The first one will explain, the first intro will explain what the tool in general is about, and then we'll run through demos of 3 use cases, after which you'll have an opportunity to unmute yourself and ask questions as well. And I would like to encourage you to ask questions in the chat function on the right panel in Hopin throughout the session, and we'll get to those questions as we find appropriate sort of gaps. And also, I would really like to encourage you to use this shared Google Doc throughout the session to sort of just tell us a little bit about yourself, what you'd like to get out of this workshop, why Scivision might or might not be of interest in your project or in your organization more broadly, to ask any questions as well, and any notes and observations. And then we can pick those up either during this hour or if there's not enough time, we'll pick them up offline later. Yeah, so that's all of the housekeeping for today. Thanks so much. Just to say, if you wish to unmute yourself, on Hopin, it's not the sort of most intuitive platform, but if you click on the orange button at the top right-hand corner of the screen, it says share video and audio. That basically, just click that, and it's a little bit like joining a Zoom room, and then you can unmute yourself and ask a question. And yeah, with that, I'd like to invite Scott Hosking to start the session. Scott's a Senior Research Fellow at the Alan Turing Institute and the head of the Artificial Intelligence Lab at the British Antarctic Survey at Cambridge University. Scott.
Thank you, Ada. Good to meet you all. Welcome to the Scivision session workshop, whatever we're going to call it. It's great to have everyone here. We've got 65 people. That's great. So we've beat my target. I was thinking we've got to beat 50. That's great. Okay, so let's give you a bit of background on who we are and where we've come from. So we're a team of scientists from lots of different domains. Sebastian, Alan, and I started at the Turing joined Turing as secondees around 2 years ago, and we're in completely different areas of science. And ordinarily, we probably would never have bumped into each other or chatted about our science. But we're all starting up our new Turing teams at the same time and hiring people. And one thing we didn't want to do was to build 3 distinct data pipelines, building a collection of data or methods and algorithms which were not interoperable with one another. So Scivision grew naturally out of that idea, and that a way to work together without duplicating effort. And so it's a tool that was originally designed by scientists and for scientists. And about 6 months after that idea popped into our heads, we started talking to Oliver, Kazra, and Ed here from the Turing's Excellent Research and Engineering group. And so they're professionalizing this toolkit now to make sure it's actually something that's usable outside and beyond just our small group. So the aim of Scivision is to help users discover data sets, not just, so you'll be probably working on your own data sets, but also to help you discover other data sets that may be out there and really help you ingest those data without having to write your own data pipeline, your own data loaders. And also to use and adopt algorithms that are out there already without having to, again, go to the GitHub repository to work out how you plug in those algorithms into your own data pipeline. So we're trying to build that glue to make it super easy to combine your data sets with your algorithm. And one thing we've done at the Turing is one of our aims is to to limit those silos between science disciplines. So let's build tools that can work right across the science and humanities remit. And we've got some really nice examples today, actually, where we've actually done that, where there've been algorithms written for humanities that are now being used for science. And we're about to go into those use cases. Now, just one last thing. So Scivision, what is it? It's a set of tools, but it's to enable people who are new to computer vision to apply algorithms to data quite simply. Or it's those people who are time poor, that just want a quick look at some data and to try something out before they, get into the weeds and write a Python notebook or script from scratch. So we've got this sort of vision, this aim to be able to do all that with around 5 lines of code. So line 1, search for your data, load it. That would be line 1 and 2. Line 3 would be search for an algorithm. Line 4, load that, and then line 5, combine those together. Now, that's a vision, and that's not always going to be the case, but it gives us an idea where we're aiming to go. And on that, I will hand over to Evie to present our first exemplar using the Scivision toolkit. Thank you, Evie.
Thanks, Scott. Okay. Can everybody see that okay?
Just needs to be full screen, Evie.
Okay, it is full screen on my screen. Sorry. Is it full screen now?
Not yet. So I saw you just press the slideshow button in the bottom right.
Yeah.
But it hasn't changed. But
okay.
That's probably okay. You can resize some things. Pull the notes down. Try again. Yeah, thank you. Try again.
Okay. Sorry.
Tamara has just put in chat there's been some issues in other sessions, so you're not alone, Evie.
Okay. Yeah, because it wouldn't let me share the entire screen.
Okay.
Just the window. But...
Should we go? We can go back to that, and as long as we can see the slide, even if it's slightly smaller, that's fine.
Is that what we'll do?
Those people watching at home, there's a full screen button in the bottom right. You'll hop in, and that will expand it so you can see better.
Okay. Yeah, let's just try this for now. So hi, I'm Evangeline Cochrane, a postdoc at the Turing working on the impact of climate change on UK agriculture and food security projects with Sebastian, as well as contributing to Scivision. And
sorry, Evie, at the moment I'm not seeing the screen at all. Is that the same for everyone else?
Oh. Okay.
Yeah, that's right. So just share it as it was. That would be fine.
Yeah.
Awesome. Thank you.
Okay. Yeah. So our use case, this slide just highlights how, in many ways, plant phenotype data is at the nexus of sort of understanding plant growth and crop growth, as the phenotype is the product of genetic and environmental inputs, including crop management and interactions between genes and environment. And therefore, phenotype data is a crucial component of models of both plant development and crops, and serves as sort of the bridge between genetic and landscape scale responses. However, traditional crop phenotyping methods are very labor intensive and time consuming, and can be subjective or destructive to plants. So in recent years, advanced imaging platforms have been developed to better understand plant phenotyping and improve models of crops and plant developments, which cover a range of scales and spectrums, as you can see here. However, analysis of these data sets remains a significant bottleneck, as they're very high throughput and hard to manually extract measurements efficiently, particularly when we're looking at dynamic traits. As part of the food security project, we've been developing tools for automated extraction of phenotype data from these high-resolution data sets in collaboration with the John Innes Centre and Rothamsted Research, and that includes this data set we have of individual images of brassica plants. We have multiple images throughout the growth cycle of the plant from multiple angles, and what we would like to do with these is look at when different plant structures emerge, like flowers or leaves, and how the composition of the plant changes over time, with two of the main challenges being that some of these structures only appear towards the end of the cycle, so an imbalance in classes could be a potential problem. Also, a lot of these structures that we'll be interested in, for instance, seed pods and branches, look very similar. So this led us to trying to apply MapReader, which is a pipeline that was developed within the Living with Machines project at the Turing, originally for historical maps, using a patch classification approach to identify railways and associated buildings. And we thought that we could potentially apply this method as it takes into account the context and the spatial arrangement of these patches to potentially distinguish between very similar looking features, and also there's a lot of really great annotation tools that help you curate a training data set of different classes. So you can see here, this is the example notebook for this MapReader plants model in Scivision, and we start by, as Scott mentioned, just loading the core functionalities of Scivision. We then search for the MapReader model, load that MapReader model. This MapReader model trained specifically on these images of plants. We then load some example data, in this case, just some data from a GitHub repository that we have set up following the Scivision template, which is described in the Scivision documentation. We then use a plant flower data source from this data set and load that data here in X-ray format, which will be compatible with the MapReader for Plants model. And you can see here one of these images that was loaded using this sort of single line Scivision load dataset process. We then perform predictions using the MapReader for Plants model, and using we specified the patch size here to match the patch size that this particular model was trained on, but that's not necessary to run this step. And you can see here that we get an output image with the flowers labeled in red and the non-flower parts of the plant labeled in yellow, and then the rest has been omitted or labeled as background, as well as the tabular sort of output for each patch, which is the prediction class label and the confidence. Important to note, this is sort of part one of a hierarchical classification process we'd like to implement. We'd further like to classify these non-flower plant patches now into leaves, branches, and seed pods. But a great part of having this model as a part of the Scivision catalog is that we can then load a different data set and see how this model performs as is on that data. So here I'm loading data from the vegetation edge detection use case that my colleague Alejandro will talk about in more detail. And you can see here that this is satellite data where they're interested in looking at coastal vegetation. And then we can just load the MapReader for Plant model again, perform model prediction on this completely different data set. And you can see that it's labeled some of these patches as non-flower plants, or the green parts of plants. You can also specify a different patch size within the Scivision API and perform prediction with that without having to retrain the model or take it to more complicated steps outside of Scivision. So that way users can sort of initially explore how they would, what might be an appropriate patch size. And I think this really highlights how Scivision can sort of be a matchmaking process. And I think that this this application of MapReader from historical maps use cases to plants is an idea that could have only really come together with the unique collaboration network that we have at the Turing. But I'm hoping as we continue Scivision development and expand the Scivision community, that's something Scivision can facilitate, is more of these cross-disciplinary applications and ideas, so that it's not necessarily to work at the Turing to look at this sort of cross-discipline application. Thank you.
Thank you, Evie. We'll go over to Alejandro now. Lots of excellent cross-disciplinary, interdisciplinary examples there. Love it.
Okay. Hi, everyone. Let me just zoom in this. Okay. Hi, everyone. So now is the turn of the environmental use cases. In this case, it's been like I am a postdoctoral research associate at Turing, and I'm working in a project about bridging scales between like ground side data and satellite data, and just to develop software for environmental modeling. I'm working with Scott, who is a leader of this project, and Ed, that also helped us to run some use cases for the environmental team. So the first use case that I would like to highlight, and it's important to know these use cases in somehow that I'm going to present, highlight how we are building the community through Scivision with not just people at the Turing, but also existing models that are out there. So the first one is the one about coastal vegetation adaptation. Maybe just some context. This research aims to detect like this vegetation coastline that you can see here in this figure, like purple one. And this is very important for ecology and remote sensing studies, because it's very challenging problem for different perspectives. And usually the conventional way is just to go to the site and collect all this data and delineate this coastline like this way. But in order to cover more areas, you can use satellite data. And here, of course, you can do this manually, but there are a lot of interest to do this more like machine learning or data-driven automatic way. So for this use case, people are aiming to find cost-effective detection of a shoreline location and dynamics. And it's here where Martin, in collaboration with Martin Rogers, who is a researcher at the British Antarctic Survey, he, as part of his PhD, he developed this model and advancing the detection of this coastal vegetation edge using like this hierarchical nested convolutional neural network. And it's very interesting, this network. Probably you can find more details in the publication, but is inspired by the BGG16 network, and essentially each of these layers, you have in total 5 outputs, and these outputs are retrieving different delineation of the coastline. But the most interesting one is the latest one, with capture most of the useful information to detect the coastline, in this case, in this sample image. And something very important to mention here is that Martin, because satellite images are very different to natural RGB images, he experimented with other bands and he found which was the most feasible combination. Here we see the outputs of the model, and in this case you can see in the confidence, it yields the confidence. So one is that it's detecting this vegetation, and here is a comparison that he has in the paper. You can go to the Martin's paper, and here you can see red values are those vegetation areas, and of course the other areas are no like vegetation coastline, and he compares with some ground truth. So how we can ingest this model into Scivision? So that's the thing that I'm gonna showcase. So in Scivision, you go to the gallery. We are using Scivision, as Evie said, to import here the functions, default catalog, low pretend model, low data sets. In this case, the model you can find, if you run this example at the BH detector, and you can find with this model which are the compatible sources. In this case, we are using some sample data set that Martin kindly provide us. And after that, you find the model in the Scivision catalog. It basically is very easy to load this model. That is a kind of model repository that Martin has in his GitHub account, and we load this model, and it was built in TensorFlow. This is just a general description of how the model loads in Scivision, which is the input and the outputs, and then the data source that we are pairing this model is, as I say, sample data of satellite images that Martin has also in the repository. In this case, it's very interesting because this image that they provide, he has different satellite images. One is planet, that is 3 meters, and the other one is RAPI, image RAPI. But here in this example, we are just using one of the examples, so we cannot very easily load one of these images. And this image, in this case, you can see that has 4 channels, and this is the size of the image, and is imported in XArray data array format. And here we see the predictions. And in this case, this is the original satellite images, and here is the prediction. It's important to know that inside this predict function, it automatically is taking the most feasible combination of bands to generate the predictions. In this case of this area, we can easily change this one by other example, just to showcase here that you can easily change this for another example. In this case, is another size. And basically, because this is a RAPI image, you can easily here just need to change here the provider, and this is gonna do the prediction here in the other area. So that's the first example, and this is one of the examples that we have attracting people out there who develop models. So I'm going to the second example. The second example that we have is the plankton image classification. This was maybe those ones that are not aware, the plankton are very important species to study ecological or climate change aspects. And in the case of the UK, we have this CFAS endeavor, that CFAS stands for the Center of Environment, Fisheries and Aquaculture Science. And inside this boat, they are collecting, and they have a kind of plankton imager who is collecting tons of images every day, and they are having surveys every year. As a result, they have plenty of diversity of images of planktons. And the aim of CFAS in this case, I will explain later, but it was part of a data study group. The aim was to develop a rapid on-the-fly methods for automatic identification of detritus. That detritus is a kind of it's an organic material. It's not like plankton species, and also classify which are the different species within those images. Here, as I say, as part of a recent data study group, Scivision was part of this data study group, the Scivision team, and they helped to prepare the data sets to be ready to analyze in this challenge. And they have almost like 60,000 images, and the participants here, they developed a model. The most feasible model was a convolutional neural network that they pre-trained using this data set, and they generate different classification levels that the data owner, in this case, CFAS, was aiming. One was zooplankton versus detritus. This is more like a genus level, that is copepod, no copepod, and detritus, and it was the finest taxonomic level that they were aiming. And this model provide really good results in comparison to a previous baseline that CFAS has for doing this real-time classification of the images. If you have any more, you want to add more details about this, the result of this study group, feel free to contact Sophie. So now I'm going to the use case. The use case is quite similar to the previous one. So here we are importing just certain libraries that we need for Scivision. In this case, we already registered this model and data set in the Scivision default catalog. And the model name is ResNet50. And basically, you can later go through this in the binder links that we have. And we load this model. Then we find the data set. In this case, is some sample data set that were used in this data study group. It was part of the whole-out data set. And CFAS kindly provide us some sample of this data set, almost 26 images. And we are going to showcase how this is imported into Scivision. This is imported in again, as array object. But the challenge here is that all these images because they have different sizes, they are concatenated into a single code information, and it contains this black background. But in the behind, what we are doing with the model is that it internally in the prediction function is cropping only the image values, and over these image values is doing the prediction of the pre-trained model. And it's the thing that we see in the final step that in this case is predicting over this data set that have this kind of format, and internally is cropping only the area of the image. And finally, we have the prediction of this model in three categories, that is copepods, no copepod, that is that one, and this other one that is detritus, that, as I say, is not of interest, but is because it's more mineral objects that you can find in the oceans. And yeah, that's it. Thank you.
Thank you, Alejandro. So if you could stop sharing your screen. So before we go to B's talk, I'm just going to quickly catch up on some of the questions, because actually I think they're all related, and it might be useful just to give you some context there before we go on to the next talk. So Katie, you asked, how does Scivision compare with computer vision in fast AI? Now, one thing I wasn't clear at the beginning, but Scivision acts as the glue to bring all the data, all the machine learning frameworks that's already out there. We're not trying to reinvent the wheel. We want to use those excellent tools and provide the plumbing so that Scivision can then plug in and use fast AI or TensorFlow or OpenCV or PyTorch, etc. So it's a way to get hold and use those excellent tools without having to learn a whole new set of frameworks each time. So one thing we've been showing today is that we've been using pre-trained models. So these models may be pre-trained by machine learning experts, and then we can use them quite easily within Scivision. Going on to Tamara's question, how much training in computer vision and machine learning do you need to apply these existing methods to your data? Would familiarity with Python be enough to get started, apply existing methods and all the data sets? So so this also goes back to what I was just saying, that if you were to build, and if you wanted to train a model from scratch, you would still need that expertise to work in TensorFlow or PyTorch or whatever tool you're using. But one thing I think Scivision can do is you could find a data set and an algorithm that may be out there within our gallery, which looks similar to what you're doing, and then that helps you build community and build that network. So you could reach out to these people and say, look, I've just found your model, it looks great. You're doing coastline detection. I've got a very similar data set. I'm trying to identify some lines or an edge in my image. Could we have a quick chat and see if there's any mileage in bringing my data to your model? So it should help. I think Scivision can give you that first step towards making those connections. Patricia, will Scivision work update channels? Absolutely. If the underlying model you're using is TensorFlow or PyTorch, whatever tool you want. So again, Scivision doesn't restrict you in how you use these tools. And then Hannah, how do you manage to isolate the plankton detritus well enough to get an ID? Okay. That might be a question for one of our researchers, Evie, B, or Alejandro. I don't know if anyone wants to come in on that one.
Maybe just to make clear, like what you mean by ID, like the final class, because it's confusing.
So how do you manage to isolate the plankton detritus? Well, so we built a classifier in this study working with CFAS that had an accuracy of around 95%. So it was able to distinguish between plankton and detritus, and not only that, within the plankton classifying sort of larger classification, we're able to drill down to different species and subspecies. But Evie, you've come in. Do you want to add to that?
Yeah, I was just going to say, if the question is about sort of how the we get these images of individual plankton and bits of detritus to start with. That's from the imaging platform that's being used, which is this plankton imager that CFAS developed, which is on this research vessel, and basically it pumps water through a very small tube and takes lots and lots of images as that water is pumped through. So because it's so thin, that sort of isolates the different particles. And they sort of developed this first step of the model, which was to crop images of individual particles through that system. So that's where, yeah, again, Scivision is sort of forming the glue between these amazing data sets and models.
Thank you. I'm going to take one more question because I feel they're going to start coming in now, but and we will we'll have these talks. So one last question from Edwin. Is the approach used in Scivision for transfer learning? No, it's for any, if you've got scientific imagery or imagery from humanities and you want to identify something or classify or extract some information or meaning from those images, then Scivision's for you. Right, B, over to you.
Hi everyone. Okay, let's try to share, because I've been having all of the technical issues today, so hopefully it's gonna let me. Yes, it is. Can you see my screen now?
Not yet.
No?
No.
Oh gosh, of course, because it would be too easy. I apologize. Should be okay. So, what's a presentation without is it working now?
No, sorry. It hasn't, you haven't gone full screen yet.
Not yet?
No.
Okay, so I think we're going to have to do this in another way. And if Alejandro can share his screen, because he had my presentation there instead of just... so sorry, guys. It's just fun. But anyway, it's a good interlude before I change drastically the topic so no one gets whiplash from this.
It's this Alejandro can share, but I can also share your cryo-EM. I can. Oh, here.
Okay.
Here we go.
We have.
Brilliant.
Thank you, Alejandro. Okay, cool. So, hi everyone. I'm B. I think that's a bit obvious by now, and I'm gonna drastically change the topic because now we're gonna I'm gonna talk about molecular biology use cases in Scivision, and you might ask, molecular, but what kind of images is that? Because now we're talking about near atomic resolution, right? So, next slide, please. This is old school when we asked to change the slide. So what is cryo-EM? So what is this that we use to see molecules? We use this thing called cryo-EM. And what is cryo-EM? So basically, when you have things that are smaller than the wavelength that the lasers would, the fluorescence imaging would capture, you actually need to think outside the box. And in this case, we use electrons. But, with great resolution comes great noise. So we actually have really noisy images when we observe these samples. And these samples are frozen. They can be proteins, they can be they can be cells, but it's just, the idea is to get the figure on the right, the protein structure, the outside, basically, the surface of the proteins from the images on the left. And yes, this is what an actually clean image looks like. And I don't know if you can see because it's very small, but each protein is one of those individual little special circles, and I have a green box around them. So now, to get the image on the right, you actually have to average over hundreds, if not thousands of those. And the trick is in how do you select and identify and actually just isolate the regions that are of interest. So next slide, please. There's also different types of sample data. So the one that I showed on the previous slide is in the category here on the left, where we have a purified sample, which means that all of the proteins in the same sample are the same proteins, and everything that is not that is to be ignored. And it's very dense, and you have a high number of molecules. But also you can have complex molecules, like, for example, filaments. And these are more complex because while on the first case, it doesn't matter really the angle that these proteins are. They have some similar shapes in all directions. Actually, in filaments, they're very different. If we look at them in this way or this way, then we are going to get different imagings because we're capturing a 2D image of 3D structures. And finally, we have the most complicated type of samples, which is when we have specific molecule detection in images that are full of other types of molecules that actually kind of look similar to the one that we want, but not really, and we're not, and we need to separate between them. So next slide, please. How do we actually tackle this problem? This is a quick slide. We can go next. We need to train models to detect these molecules, right? And we need a lot of data, because everyone that knows machine learning knows we need a lot of data. And we need, and with this data, we can have a reliable, accurate model. But this is where Scivision comes in, because it can enable us using the existing data sets on new models, or, applying existing models to new data sets. But in this case, particularly of the molecular things, what type of data sets are we talking about? Please, next slide. So all of the data that exists in cryo-EM, the raw data is uploaded to this one database, like the Empire. It's like the IMDb, but for really gray images and doesn't have a score. So a less cool IMDb. And all of these data sets are already there. It's an entire community. And if we have, with Scivision, just this one line that we could access any of the data sets, we have access to this entire world of gigabytes and terabytes of data. And it's really big. Like, we struggled to find a small, a data set that was small enough that would be okay for a notebook to show. So that's for you to see. And then there are some problems, right? Because these data sets are too big sometimes. And actually, we're very reliant on humans annotating it by hand. Yes, someone has the job. There's a poor PhD student that has to go by hand and isolating all of these things and getting them all to be average over the end. And again, very highly reliable. So for that, next slide, please. We have synthetic data sets, which have the caveat that it's not real data, obviously. But in reality, we have the ground truth of everything that we see. So we know what, in this case, we have an alphabet soup. So and each letter it's as if it's a different molecule. And basically we have, we know the positions, we know the orientations, and we know the identity of each of these molecules in the image. We then put some noise in it to mimic the microscope optics and all of the conditions, and we get a very simple first prototype of this. And after that, we apply a method, a model we called Odin, to detect all of these molecules in the image. So as you can see, the detections are everything that we see on the image. So I will show you now the notebook. The next slide is the link to the notebook. I don't know if it's enough time for everyone to see, but I can share the link quickly. And if we open, thank you, Alejandro, you're a great assistant. If research doesn't work, this is great. So in this, we have a little bit more than 5 lines because we have two data sets, right? And the idea is that we import the model and we just prepare everything, and if we scroll, yes, we just load the pre-trained model that we have trained on the synthetic data set, and then we load the synthetic data set. And as you can see, it's literally, we can generate the images on the fly. So what I forgot to say about the synthetic data set is that it doesn't get images from anywhere. It actually makes the images as we need it. So we have an infinite data set, if we want to, of randomly generated images where we know the exact ground truth. So this can be used to train models that need detections and all of that, which is what we did. So this is the example of an image that has been processed, and it has the beautiful EM filter. And then when we run the model through it, if you scroll down, please, we see in purple/maroon, I hope everyone can see, that's the boxes that were actually detected. And you can see that it was pretty decent detection. But now, what about how do we use real data? So if we go down, and I said we can access the database, we see here that to access the database, you literally just need one line. You need to say what type of entry do you want from this database, and then you load it through Scivision, and you can show the image right away. So this is an image, but you have to see that this image hasn't had any normalization and anything. So when we can actually run the model runs on it, but we don't have any bounding boxes, but yet, because then we can just work on it. So that's everything for me, and thank you.
Thank you, B. Fantastic. Really great talk, especially considering all the tech issues you've experienced so far today. Evie, did you have a wrap-up slide as we move things around slightly?
Yeah, so I just want to let everybody know that all of these example notebooks are available on the Scivision Gallery GitHub, which I believe has already been linked into the chat, but the link is also here. So yeah, github.com/Scivision Gallery, and you can look at all these notebooks in more detail, run them yourselves. And thank you so much.
Fantastic. And I cannot see Ada here, so I was going to hand over, but yeah, she's coming back in. So Ada, we're back.
Hi, so yeah, very well done everybody on the presentation. So now we'd just like to take some questions from the audience, and thanks so much for adding questions in the chat as we were listening to the presentations. And I think some of these were picked up after Alejandro's part. So just you will have to help me to see how far we've actually answered them. There was a question from Santiago around model robustness and explainability in Scivision. Scott, was that picked up?
I think that Ed picked that one up.
Oh, great. Okay, so that was. So then, so that's great. So we've cleared up the chat. So can we now invite more questions from the audience? And you can either type it up in the chat, or you can click on this orange button at the top right corner of the screen that says request video audio access, which will allow you to share your video and unmute yourself and ask a question that way. So it'd be great to have some discussion. It doesn't have to be a sort of an outright question. It can be a kind of I use this so far. Would Scivision enhance some aspect of, whatever you need in your workflow? So it could be a bit more of a discussion, or you telling us what you're working on, and if you think there could be some potential for collaborating or testing Scivision or anything like that. So I'll leave it there, give you a few minutes to think if you'd like to ask any questions. We've got Andrew Smith and Fatma who've requested access. I don't know if this is an old request or a new one. I'm going to approve it and then see if you wanted to unmute yourself. Don't feel any pressure if it's an old request, then you can just ignore it.
We can all just awkwardly stare at each other now for the next 15 minutes if needed.
I guess there is one question in the chat. Emily Lines asks if there is really high reliance on label data, which, as we say, can be very time consuming to create. Are there any community efforts to share label data? Yes, it's in the chat.
I didn't see that question.
It's in the event tab. That
Event.
was a good shout.
Yeah.
Alejandro. Yeah.
Yeah, maybe B, if you want to.
So that is that's why we actually made the synthetic data set, because we struggled a lot. And we're talking about really big, large data sets that were annotated by hand. And because it takes so long to annotate by hand, people tend to not be, at least in the field, tend to not be very comfortable sharing all of this work so that, other people then use. So I'm not sure if there's any community efforts in the specific field that I'm working on, but there are efforts to automate and make it a better, easier annotation rather than just being so reliant on humans.
Can I also add as well, in the sort of Scivision catalog, one of the fields that we've been included so far is, for data sets whether labels are available, and for models whether labels are required, which helps you sort of identify matches between models and data sets that way. So hopefully as the catalog expands, that will be a resource for users to identify labeled data that already exists as well for different fields.
Thank you. And we've got a question from Patricia. She's asking which ML techniques are being integrated and how are they tested for accuracy?
I wonder whether that's one for the Reg team.
Sure, I can give a brief answer. So the in terms of what's being integrated into Scivision, really the sky's the limit. So we're aiming to create this community of contributors, of anyone who works on any kind of computer vision algorithm or model. So they could be using any conceivable technique. And we have this submission process to include models and indeed data sets in what we're calling the Scivision catalog, which is our sort of repository or database of both computer vision algorithms and data sets that they could be run on, so image data sets from the different fields like the ones we've seen in the presentations today. So if you're interested, Patricia, I'd recommend to check out the sort of Scivision documentation, which is linked in the doc that Ada has shared in the pinned message in the chat. But essentially, there's a process by which we take submissions for new models to be included in that catalog. In terms of this is related to the question that I answered for someone in the chat before. Are the models tested in any way? At the moment, the answer is no. So we plan perhaps in the next sort of phase of Scivision, one of the next features we may work on will be some kind of process to evaluate whether the kinds of submissions we might be getting in terms of computer vision algorithms to the Scivision catalog are in some way good or accurate or have some kind of metrics by which to measure them. At the moment, that's not something we've worked on yet. We've been sort of working on these example use cases you've seen in the presentations today and sort of coming up with the idea of the catalog in the first place and getting started with building a platform that can link dataset providers and model developers. But it's a good point. If anyone else wants to say anything about that.
Thanks, Ed. I guess we can move on to the next question from Mikhail. If I'm reading this right, did you use internal external cohorts to test the adaptation of the techniques in unseen cohorts? Don't know who wants to take that.
Is that directed to a specific talk? I don't quite understand what cohorts are. I don't know. One of the presenters sees the connection to their work. Maybe we could, yeah.
It's for B. Yeah, okay.
I'm not sure I understand the question. I'm sorry. So you mean, like, for example, because we have access to all of the data sets, whether we test on different ones, and yes, we test on different ones. Obviously, if the images are bigger, it takes longer, but it loads the images and it deals with the file types that are very specific to the field. I don't know if that answers your question. I hope it does.
Mikhail, you can also click on this orange button that will allow you to ask the question kind of verbally, which might be a bit easier because you can then kind of have a discussion with me and explain if you wish. While we're waiting, perhaps it might be worth to moving on to James's question, which is for Alejandro. So the ID went to a low level, i.e. copepod. Is it even possible to consider species identification given their small size, and how many characters or characteristics would be needed? Is there an example in another system that has been done on these kinds of tiny invertebrates? So that's for you, Alejandro.
Okay, maybe James, thanks for the good question. I guess you refer to that's why as part of the data study group we explore different level of classification, and the example there that we have in the Scivision notebook is only referring to one of the models that was trained to classify very general level copepod, no copepod detritus, but we have other models that at the moment CFAS they have this model, but they are classifying a finer taxonomic level that you mean in this case species. I guess so far, I guess this model was trained only to detect over plankton images. I'm not sure if this model could work in another like different tiny invertebrates as you say, but maybe it's just an example that in this case you can call by Scivision a model that was trained to classify copepod, no copepod, and maybe in the future you can use these features that were to extract these particular images and retrain over other set of images of another animal species, whatever. But at the moment, it's possible in this case through Scivision run different models that are classifying different targets. I hope that it answers the question. And about the characters, maybe you refer to the features. In this case, was a neural convolutional neural network, and it was only using the RGB channels of the images. So in this case, the features are extracted from the in the model. So we this traditionally they were training a random forest that were extracting many features of the images, but it is very time consuming. That's why, as part of the data study group, they developed this new framework using ResNet model that allow to extract the automatically the most important features from the images. Yeah, that's it.
Thank you, Alejandro, and thank you, James. I can't see any more questions coming in the chat, so perhaps I'll give a minute to any last questions that you might have. Otherwise, otherwise we could all go to have a sort of early lunch, get ahead in the queue.
I noticed Mikhail's requested to.
Oh, perfect. Wonderful. Let's see if Mikhail can now unmute himself.
I think he already answered. He already asked his question, didn't he? So.
Yeah, it looks like Mikhail was approved to join the session room, but then sort of got a little bit lost in this limbo between the audience and the session. Hopping is just wonderfully surprising when you try and manage an event. So thank you all for bearing with us. And sorry about the technical glitches, and be sorry, it's been difficult to share your screen. I heard some people saying it depends on whether they're using the latest version of the browser or the latest version of the operating system, and it's just, it can be quite chaotic. Thank you, everybody. If we don't have any further questions, let's go have lunch. And if you have any sort of further questions for the team, please feel free to interact via contacting any of them really directly, or the RAM team if you're wondering about potentially even testing the use of Scivision in your organization. So I'm just writing the email address of the RAM team. And all partnerships, it's all in the shared Google Doc. You have some contacts there. So yeah, try it on GitHub, and if you have any questions, come back to us. And thanks again.